Welcome to our Friday webinar. Today we're going to be talking about gene errors in Excel, and this is in some ways really, I guess more generally could be described as talking about errors with dates coming, or the date field in Excel, when you have data that you don't want to be a date and it gets interpreted as a date. And I guess I shouldn't pick on just Excel. This is also an issue that happens in Google Sheets. And there was recently a study conducted looking specifically at genomic data and the number of errors that you would find. And this was something that actually was pointed to a user pointed me to this most recent article. I had not seen it yet. And often in past webinars, I've talked about the date field in DataGraph and how it's handled differently than in other data management types of software, which I will talk specifically about for sure. But so this user pointed me to this article, which was talking about these gene errors that, again, I sort of mentioned this in the past, that this is not a problem you would have with DataGraph, and that there was a study done years ago that showed that about 20% of Excel files posted online that were shared as supplemental information for articles had errors in the gene names. And this most recent study reevaluated that and found that of the files that they looked at, 30% of those had errors in the names. And this was maybe surprising initially, because there had been a big effort in terms of education and trying to let people know that this error occurs, and yet it still is very pervasive. In fact, some of the gene names have been modified in order to prevent this error from happening. So anyway, so this is an issue that I think is worth talking about. And I thought it was very interesting to look at some of this data, and hopefully you will as well. I do want to say that I am not a biologist. So my background, and I guess I often don't introduce myself. So any of you who are new to coming to these webinars, my name is Pamela Schultz, and my background is in chemistry and environmental engineering. And so please don't mistake me for a biologist in terms of presenting this information. And I really actually am hoping that one of the things that can come out of this webinar is a bit of a dialogue with some of you who potentially work with data like this, in terms of understanding the workflow that you use and how DataGraph can be helped to solve this problem. So that's what we're going to talk about. And in particular, I want to show you a kind of a mini case study that we've done to look at how DataGraph handles this data. Again, these types of errors are well documented for genomics data, but they can happen with any type of data. I've certainly had that happen to me, where I've had a cell in a spreadsheet that I did not want to be a date, and suddenly it gets converted to a date, and you just don't see or realize it later and don't seem to be able to get it back again. And we're going to make some graphs. So for any of you who just want to get more exposure to the program in terms of bringing in data, manipulating data, and just creating some nice looking graphs, we will certainly try to do that as well. Before we do that, however, I want to show you something new this week. In fact, we just uploaded this in the beta, I think this morning. So for any of you who are beta users, what I'd actually like to do is share my screen, but also to show you how to use the beta if you've never done this before. I have in my downloads the latest version of DataGraph beta. I work primarily in the beta because that's what we're doing. We're constantly trying to improve the program, as well as just, if there's a small bug here and there, or fixes, we're always using the beta. So if you download the beta from our website and you just go to the DataGraph website, go to the downloads page, you will see there's a link for the DataGraph beta. If you're only using the release version, that's completely fine. But obviously the beta version is something that is, we like to promote it because it gives us flexibility. It lets you see the latest things and test out the program prior to us having a new release and a new update. So when you open the beta folder, then there's a text file in here that will show you a list of changes and the dates that those have been made. And then there's also the application itself and a link here to, actually let me open my annotation software here. Okay. Yeah, so now I can point to things. Okay, great. There's this link to the applications folder, so you can drag the application into the folder here. I just typically always do that. Then it's popping up on my other screen, so I'm dragging it over. I get a message, do you want to overwrite this? And I will say replace. Now I'm doing this live. Hopefully I don't regret this and everything, and the new beta is working fine. Anyway, so now I can go down to where I already have a link to DataGraph on my toolbar. It's going to ask me if I want to open this. I say yes, and great, there it is. So now I have my new version of DataGraph. And what I want to show you is actually a request for a feature that we had just recently. And so I'm in the example files, and this is related to the animation of data. And so let's see quickly, maybe we'll just do a very simple one here. So I'm searching on animations, and just to illustrate this, I'm going to open this really simple one, animate a point along a line, so that you can see what this is all about. So here's my file, and let me move this out of the way. Okay, so let's make these points bigger and red, just so you can see. So the animation variable, if you've never done this before, if you've never used animation, the controls are down here at the bottom left. And essentially, this is a built-in variable that has a set duration and a range that you can change. And the value of the variable is shown here in the bottom left. So when you hit play, actually, I should have my mouse. So anyway, you can see how this is playing right now. Oh, I can also loop this so that can just play. You see that this has two different points that are moving, actually, I guess at different speeds here. I just wanted my mouse to be, there we go. So now you can see my clicks. So what we did, there was a request to change or to add the ability for the duration to be a variable. And this was something that at first we were sort of hesitant about because of the fact that you could have potentially some feedback loops that wouldn't make sense. However, what's interesting about this, for those of you who are doing animations or are interested in animations, is that you can actually then have the duration depend on the value of the animation variable itself. So here I have the duration is 10 seconds, which means that it takes 10 seconds for the movie to go from beginning to end. But I can actually then make this dependent on the value of t. And when you do this, the impact, let me actually just stop it and we'll go back to the beginning, is that you have the animation doesn't play continually. It will change from being very fast in the beginning to very slow at the end. So now this is going to go much, 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 much slower. This is something actually in the past I've used with some COVID data actually to show kind of the initial time periods going very, very fast and then having later time periods go much slower. That was actually a lot of work to get that to change the rate that my animation went. So this is something I'm looking forward to trying out, and I really wanted to show you. Okay, so that's that update. Let's go ahead now and get to the main part of what we're going to talk about today. From last August, autocorrect errors in Excel still creating genomics headache. There were a couple of things about this article that struck me in particular. And one of them, we've already talked about the fact that this appears to be a growing problem. That's what's documented here. They have this section here, avoid or adapt, meaning, you know, if you continue to use spreadsheets, you're going to continue to have problems. And the solution being given, at least for some computational biologists, to use scripted computer languages such as Python and R. And the quote that I thought, I guess not a direct quote, but the comment that I thought was really interesting was the comment that, you know, she just doesn't have time for learning a programming language. And I know our users are a mix of people who some definitely do programming, but many of us don't necessarily have time to learn the ins and outs of programming. This is one of the reasons I think DataGraph is sort of interesting in terms of being used partly for this purpose, because it's not going to take you anywhere near the amount of time to learn how to use DataGraph as compared to Python or R. I wanted to look more closely also at really the research article that's the subject of this news article. And there's it's reference number 3 down here. If you go to that link, the name of that article is Gene Name Errors, Lessons Not Learned. And again, I'm going to scroll through this a little bit. I'm not going to look at everything here, but there are a couple of things that I thought were particularly interesting. For example, the results of testing various software. And they looked at Excel, Google Sheets, LibreOffice, and actually Numeric. That one I was not aware of that one before. And what they found was that both Excel and Google Sheets had this problem consistently, both with opening data, pasting data, and with typing data, that they converted the data into dates when it was not supposed to be. That actually does not happen with these other, and I think both of these are open source spreadsheet programs. In any case, this is the subject of what we're talking about today. What else is interesting here? This, maybe not this one so much. This is talking just about the some counts of the data. This table I thought was interesting because it shows you the over time, the number of errors. So they screened publications over all these years. And again, you can see the percentage of various, how many articles they found that had errors, or not how many articles, how many spreadsheets posted with gene data in them had detectable errors. And let's just go down to, so again, I won't go through all this, obviously, but I want to show you one of the ways that they tested their software. And this is what I also did with DataGraph. They have a set of strings that they specifically came up with to use for testing how the software reacted to bringing this data in. So basically, you can test this out for yourself, see if you see this problem. I actually don't have Excel on my computer, so I've been using Google Sheets to test this out. But if you just take this data and copy, and here's a sheet where I've already done this, but you can see me paste this in. then one thing that you'll notice right away is how some of the entries are right justified. So that indicates that they're being interpreted as a number or date. And the left justified entries here are entered in as text. If you double click on any of the entries that are on the right, you'll see this date field or calendar pop up, indicating that by pasting it in, this has been interpreted as a date. And again, this is one of the tests that they did during this case study with these other software tools to see how they would react to bringing in the data in. So let me talk now about what we did with DataGraph. And I actually have a document. There we go. That I've been putting together that talks about these 3 different tests that we did to kind of assess how DataGraph would handle this data. So the first test that I'll show you is just importing the test data itself. The second test was to examine corrupted data. And the third test was to look at exported data. So the first test, to just do this quickly, you can see here's the test strings as we talked about. And then when you paste them in, DataGraph doesn't corrupt the data. And I think you all kind of know that that's kind of the punchline here. But let me show you if I go directly from the website, what happens. So I'll copy this. And I now go into DataGraph. And let's make a new file. Okay, so here's how it gets pasted in. Now, first of all, sometimes data from the web comes in a little funny when it's, HTML formatted. So it's adding spaces in between each of the row entries. There's a handy shortcut for removing those where I can just right click and say select empty rows. And that will select all of these rows in between. And then I hit the delete key. And now I get the data in the way that I wanted it, which was, just showing me each entry as an individual row. Now, when you bring data into DataGraph, it also needs to interpret what data type it is, because DataGraph has 3 data types primarily: numbers, text, and date. Although you could argue date and numbers are really also, that dates are also numbers. We'll talk about that in a minute. But the interpretation is made for the entire column. This is one of the big differences between DataGraph and spreadsheets, is that in a spreadsheet, the interpretation of what data type a given cell is done on an individual cell basis. This is why you have a single cell within the sheet being converted to a date versus a non-date. And there's a question about providing the slides. If you mean the document that I was showing you, then absolutely, yes, that's definitely something that we're going to post that online. So what DataGraph is doing again is it is interpreting the entire column to try and determine what type it is. And when it's doing that, it's often looking at the first few entries within a column of data to try and discern what is the data type. Here, what DataGraph actually did was, when I pasted this in, it said, these look like numbers to me. And as a result, you can see how the data from rows 4 to 7 is showing up as red. It's because it's assigned this a numeric column, but these are not numeric values. So it's just letting you know there's an error there. And I find it very handy that this built-in error checking and the ability to just see very clearly that, those are red, it's not being understood. Now, sometimes that's not a problem, depending on what you're doing. But for this, we actually want this whole column to be interpreted as text. This is not, these aren't numbers. These are not dates. These are, in fact, ways of referring to individual genes. So in DataGraph, many of you are probably aware of this, but for those who haven't done this before or are new, we have all these gear menus throughout the program that make it easy to do all sorts of different shortcuts. And one of those is to change the data type for a column. So I can convert this to text. I could convert it to date. I could also convert it to a binary number. That's probably not something that most people will need, but that is there. You also can access the same menu by just right-clicking on the column itself, and you'll see again, convert to text and date. I'm going to convert this to a text. When you do that, you see there is a little AB in the corner here, also changed my icon here, but it didn't actually change any of the content of my data. This is one of the things, again, that's very different with DataGraph. It won't change the underlying information. It's just interpreting it in a different way. And as a result, there's no errors because just any of this could be text labels. You could type anything in here. It's not going to be wrong. So here I could also just, this is my test strings. Now, if I converted this to a date, just to show you what would happen, I can do that as well. I can say, make this a date. The program, again, it's showing errors here because these are different formats. Even if these were all dates, they would need to have a consistent format in order to be interpreted. And the format of a date string in DataGraph is what you can specify by going into the object itself that represents this column. And you'll see how there is a list of all different format types that are built in. There's also a way to specify what's called an ICU format. So if you have a very strange way that your date is organized, there's very few times when I have had trouble bringing in dates and have them not be interpreted or find a way to interpret them. I'm not really going to go into that here, but what I would do is encourage you to, you can, click on the little question mark there, or just use the gear menu, go to the help and go to the documentation on the date column. You can find out more about that. Okay, so again, I can convert this back to a text. There's no problem with this. It's not changing the underlying information. You also notice when I click on it, you don't see, there's no change of what's in there. I guess maybe just to give a little bit more information about dates before we move on to the next part of the test. DataGraph has, actually expand this out here, a built-in date format that when you do have dates, that DataGraph uses to save the dates when it's actually saving the information on your computer. And that format is, I think I'll actually just put a text label here and make this very large to type this in. That format would be year, colon, month, colon, day. There we go. So the way that I have this written is, again, the way that DataGraph is saving date information on your computer. So when you even look, let's just make a brand new date column. I can do that using the Command 4 shortcut. Here's my column here. Now the default is month, day, year, but you'll see, actually here it is. There's the DG format. This is one of the formats here. And it even, if you convert to that, it will show you all the different options for how DataGraph will interpret dates that you put in the DataGraph format. But just to, make it simple. So if I type 2020:0101, that's not going to show up as red. Now, if I typed December 1st, is that going to be interpreted? No, it's not a cell by cell basis. I've defined the format for my date column for this particular date column over here. So other dates would also need to be formatted in this way. And then they would work fine. So a question, are you saying that DataGraph keeps the original strings entered and makes interpretive changes for display and calculation, which doesn't alter or lose the original input text? I clearly could not have said that better myself. That's exactly what I'm trying to communicate, that the underlying string is saved, and then we can display that in different ways. For example, there is actually a display option here that would allow you to just show the month and the day if that's all I wanted to see. Now, again, when I type this in, I'm going to type, I don't know, there we go. That's about, I keep doing the first. Let me do something else other than January 1st. But I could also actually even show you some, no, I'll just do this one, the month day. If I wanted to display the dates like this, there we go, my date column, then I can display it like that, but the underlying format is still what I originally typed in. Now, again, I could still have typed in formats like the format you see over here and change the display. So it's not just that I can do this with the DataGraph format. If I convert this to a date column, again, I have some that are not in the right format as defined over here, so they don't get shown, but I could display this as well as month, day, year. And then how I type it in is defined through this here. But again, I guess, so it's almost like there's three things. There's the way it's saved on your computer, there's what you're typing, and there's what's actually being displayed. And let's see, here's I have problems when I plot climatologies, which are averaged month or dates, but are not associated with a particular year. I usually get it to work after much clicking, but do not totally understand how to do that correctly. That is that's definitely a format that we can handle. And I think there's honestly probably a couple of days to do this. So if you have dates that are just let's see, you have averaged months or year, average months and dates, but no year in it. So one way that I've done that before is by using fixed year as a format. So if you have, let's see if I maybe just make a new date column. There we go. And I'll change this to fixed year. And actually, you can see here's some suggested formats that would work with this. So if I want to type in my data, or the data from my file is the format month and day, and again, it has a fixed year, then I can actually type in what year this is. So if this was 2020, I could say the year is 2020. So even if you're not really using the year, it can you can enter that by having you can put in any year here basically, and it will still organize and interpret this properly as a date. So again, if I made 4/09, now this is going to be April 2009, and I could display the month day in various ways. If I added the year, then it's going to show that as well, but I could have the display data like that. So fixed year is one way. I think fixed day is another way. If there's if you have a string that's not, you're having trouble with, definitely let us know. But I think fixed year is probably one of the handiest ways to use that, to do that, to make that work. Okay, let's continue a little bit. I know there's one question on dates, and I will try and get back to that. But I want to continue a little bit with going through what we've been working on in terms of testing how DataGraph handles this data and how this compares to, again, spreadsheets. So here's just a quick example where, again, you see this is what I typed into Google, or pasted into Google Sheet, and if you double click on it, it's being interpreted as a date. So the next thing that I wanted to do was to look at what happens when we examine data that we know is corrupted. And this is a screenshot of taking data that was in a Google Sheet, and I exported that into an Excel file. And then I opened that Excel file in DataGraph. And this is what it looks like from Excel. And you can see how all the cells that were being interpreted as dates show up now as a date format, as the native DataGraph date format. When DataGraph is importing data from an Excel sheet, it checks, again, it's trying to check it has to check the overall column to try and identify a type, but it's doing that by looking at what is in those strings, what is in those cells. And when DataGraph sees an individual cell from Excel that is encoded as a date, DataGraph imports that and gives it its native date format, which you see here as these, again, the year, the colon, the month, and the day. So that's why you see it like this when it's imported. This is different than when Excel saves a date, it saves, and I cannot maybe remember the exact definition, but it's something like the number of days. Maybe it's the number of days since January 1st, 1970. I really should check that and know, but it's a number that's around, they seem to be around 40,000 when we have these errors. So if I go back to this study here, what they did to identify these errors is they looked for, let's see if they show an example here. I thought they did. They looked for errors that had a particular numeric value. So they looked at the values of cells, and when those values were in a certain, at a certain point, maybe I can't find this quickly, and I don't want to spend a lot of time on this. Yeah, so basically they what they did, or their computer program does, and actually I should just go to this website, and let me put this website on the chat as well. So this is the work that they've been doing. They actually have this continuing project where every month they check and see, look at articles that have been published, and they run a screen, And then they output on a website all of the papers that they identified with errors in them, and where the names of those spreadsheets that have the errors, and then they aggregate that data to how many errors they found. And this link that I sent you on the chat is the result of the scan that they did for August of this past year. And they show you, this is based on a script that they run. So you can see the script here. And actually maybe this will show what I was trying to illustrate with the numbers. So yeah, here's the section. Look at the errors detected. And they show you a line here that shows this is the ID for the article. This is the name of the spreadsheet that has the error. They show you the species, the number of errors, and then the two numbers that you see after that are the numbers that they suspect are incorrect encoding of data into dates, where the data has been converted to a date. And so they're really looking at the numeric value. And that's one thing I think that's interesting with DataGraph, is that when in DataGraph brings this data in, you don't have to say, oh, do I have a number that's 44,257? We can identify these errors just by looking at what's in the cell. If you see something that has the colons in them, then that is that's potentially one of the errors, because it's a cell that's been converted to date format. Let me go ahead and show you, in fact, let's see. I wanted to go to, here's the errors. And I apologize for having to scroll around. I had all these links all set up on my computer, and since I had to reboot, I should have figured out how to have Safari save that for me, but it didn't do it. Okay, so journals affected. Again, they aggregate all this data. Okay, so here's their paper of the month winners. These are the papers that had the most errors in them. And the paper that we want to look at, oh, actually, that's right. I have my DataGraph file that should have this link, so I don't have to search. Here we go. Yeah, this is it. Okay, great. Okay, this is a file that where I've been sort of playing with this data a bit and looking at this data. Before we get into this, though, let me show you the. This is what I wanted to do. Go to this link, and if any of you want to follow along and kind of make this graph with me, or practice kind of bringing this data in, I'm going to post this on the chat. So this is a paper that they identified as having a lot of potential errors. That's one of the things I should say, is that they do make it clear that to really be certain that there is an error, their software or their script is detecting likely errors, but you'd want to go back and verify that that is in fact the case. Anyway, the link that I just put on the chat is to this paper here, which was identified, again, through their process. So this is not something I identified. This is something that was identified through this script. And in particular, one of the tables that they, oops, that's not what I meant to do. One of the paper or the Excel spreadsheets that they identify is this one, table 7 Excel SX. So if you want to go ahead and download this table, actually, this is one of the larger ones. Maybe I should have picked one of the smaller ones, but this is the one that had the most errors detected. So I thought this would be kind of an interesting one to look at. And, oh, it's not in my downloads folder anymore. Let's just see if I can do this quickly. There we go. So I've downloaded this one, again, table 7. If you want to, go ahead, download it on your own. And what I do lately when I download a file like this is it will go into the downloads folder. I'm clicking on this little arrow here to see what I just downloaded. And then instead of double clicking the file here to open it, although you can have, if you haven't done this yet, and actually I don't have it yet on my computer, you can have DataGraph be your default program for opening Excel files. But what I have been doing lately is just showing the file in the Finder. Then that gives me the option of right-clicking on it, saying open with, and I can just open with DataGraph app. And it takes a moment because this is a big file. Okay, spinning wheel. Don't take too long. There we go. It's always that fear that it's just going to keep spinning, right? Okay, so it brought in this data, and what do we have here? Okay, we have the data when I just said open with DataGraph, and so it's opened it all as one flat sheet. I don't know if this was actually in 2 different tabs within the original file, but you see how there are, clearly there's a section here that has names and symbols that we would want to separate from this data itself. This has 438,000 genes in this set. So the first thing I would do here is just place these 2 columns in their own group. So I can do that, highlight those 2 columns in the column list, place those in a group, and I'll do the same for all the rest of the columns that I have here, put those in their own group. Now, one thing that DataGraph always does is it will take the first row of data and try and use that. Well, if it looks appropriate, it will try and use that as a header string. And it did that here, but this really isn't what I want as my header string. What I can do is just double click this. I'm going to remove that as the header for this first column and place that actually in a note field. So I'm going to click on that note field, and we'll just paste that there so you can see what was in it. Actually, that's a lot of information in that one string. I didn't even realize that there was that much in there. But we can also, we can put this in a pop-up note. It's maybe even a little long for that. I'm just going to place this note actually in a group itself, and then I can just hide that from view. Now I have my 2 groups of data. I'm going to hide the second group and click on the first column, first row, right click and say use row for column titles. Now those are all populated nicely for me, and I can delete that row. Don't let this pop-up throw you off when you're using the program. This is meant to save you from making a mistake. It's asking, do you want to delete, because I hit the delete key, do you want to delete across all the columns, only visible or only within this group? And either only visible or only group would actually work in that case. So this is my data, and this is the set of data that, again, I can do the same thing. I can highlight these rows and delete them only in this group. And now I also have this, these are my columns, column info or something like that. Can also just put this in this group so that it can be the data can all be together. If I'm going sort of fast and you're not used to using groups, I have groups nested in groups. I do that all the time. I find that to be very handy. So what I wanted to do here was to show you how I would evaluate this data. So I have 438,000 rows of data, and I want to detect where is there an error in these within these genes. So within the data table, I can do a find. So I'm going to do Command F, and that will pop up this bar here that allows you to search through the data table in a lot of different ways. Actually, you can do numerical values, you can search for text. By default, it will search everything in the table, but you can also specify individual columns. Just like the menu selectors, it will show things nested in groups. You can see this looks familiar to you if you used to selecting columns when you're actually plotting data. You can also, this is actually sort of cute, if there's one particular column that you want to make sure you're only searching in that column, here I actually wouldn't need to do this, but just to demonstrate that, you can click and drag the column header and drop it, since it's a menu selector, just like in the commands. And now I would be searching only this column gene for when text contains, and I want to search for when text contains colon, and that's it. Because again, what DataGraph has done is it's checked each one of those cells, it's looked to see what's in them, and any cell that's been encoded as a date, it's going to be in this date format that we talked about earlier, the year, the month, and the day. And when I do this find, you can see down at the bottom here that this identified 1,234. I just think it's kind of cool that the numbers 1, 2, 3, 4. That's the number of errors identified in this data set. And in the interest of time, I won't go back and show you explicitly, but if you go back and look at that web page where they did this analysis, that's exactly the number of errors that their script also found. But again, when it's listing the errors, it shows them in those strings of 40,000 blah blah blah. So I think this is really nice because this is a much more it's not only showing you that it's helping you to identify that it's an error, but it's even showing you what date the error is, what it was interpreted as. And there's certain dates that are notorious for being gene names that have been converted into errors, like September 1st, for example, September 6th. You see the same dates repeated over and over again because these are probably the same genes that have been misidentified as dates. So that's one thing. Another thing that I did in terms of visualizing this data, or tried to think about how would I visualize this data, is I made a plot of these two columns of all these genes. And first of all, if I just do this and hit the space bar, you can see how, again, there's a lot of genes. So because this is over, I think it's 100,000 is the limit that I have set up, then individual points will just show up as pixels. So we're not, although this is plotting a relationship, that's not really what I want to show here, but you see there's a nice distribution. These are a percentage data of maybe activated genes. Again, this is definitely not my particular field, but the reason I like this was because it showed the genes kind of spread out all across this image, or represented kind of spread out across the image, and then to use this as use that image to help us to visualize the number of errors occurring. So here I highlighted my columns. I just hit the plot command to plot these. Again, it's not, even though the marker style is a filled point, because there's so many points, it's switching it to pixels. Otherwise, this would take a really long time to draw. So it's much better that the program has this way of just saying, yes, there's way too many points to draw individual ones. I'm going to switch it to pixels. And in terms of saying, okay, well, how can we within this, within my graphic here, show that show which ones are corrupted, then let's just go ahead and make a copy of this command. And I'll do this with the option drag to make a clone. And for this one, I will create a mask where I'm going to say when the gene, let's see, matches. There we go. So I typed in, I don't know how easy this is for you to see, but I typed in a logical statement where I said, if the gene matches star colon star, so it's identifying every row where the gene name has a colon in it. And these are all of the genes that have, again, that are identified. I'm trying to have let me actually change the background color here. Oops. To show this a little bit clearer, I want to change the color of this one to something, make that gray. And we'll make the points, we could just make them black for now. That's actually very, very easy to see. So then you can see now that I've used the mask to filter in on the 1,234 rows that have these errors in them. Now the points command that I have set up here will switch to drawing the actual individual point rather than drawing it as a pixel, because I've not exceeded, again, some very high limit of points. And here you can see them. I mean, some of them are overlapping, but you can see the individual points. I could change this, maybe even have a bit of an outline here to maybe so that when they were overlapping, you could see them a bit clearer. In any case, I think this is kind of an interesting representation to see where all these errors are located, or to get a visual that represents those potential problems. Another thing that's very handy. So say you wanted to have now you want to count the number of errors. Then what you can do anytime you set up a mask is you can extract a column that gives you a binary, either a 1 or a 0, depending on whether that row qualified to be drawn, whether it satisfied the criteria. So again, I just, so you can follow along, I hit the gear menu, I said extract as column, and I'm selecting mask. So any command that I think, actually I say that and then I wonder, does everyone have this? Maybe I'm not entirely sure, but certainly for the points command, you have this option of just extracting this out. This creates now a from command column that is based on that representation of the data. And you'll notice that the genes where the numbers do not appear to be corrupted, these are clearly not, that's not a date. These are showing a value of 0, indicating that they did not satisfy the criteria to be drawn for that mask. If I go back and do my command F to find, and it will remember the search that I did last time. So now it is just pulling in or showing me a reduced table that just has the genes with the error. And sure enough, the mask value there is 1. Now to quickly, this is the last thing I'll do, and then we'll see what questions people may have. To quickly do a count on, well, how many errors are there versus not there. Now, again, I can do it by doing the command F, but that's not really documenting it long-term, or that's dependent on me doing that to see the number. One thing I can do is actually just hit the space bar. And since it's everywhere where there's a 1, that's where it will show it's doing a histogram of the values, and it also gives me a sum. So again, you see that 1, 2, 3, 4 there. And then finally, what I can do is add a new graph and then just add a pivot. And this is a quick way to, you can see within the pivot table, there's a summation of both the number of zeros and the number of ones. And then at the bottom of the pivot command, I can just say that I want to display a label there. And sure enough, there's my, oops, try that again. There we go. There's my 1, 2, 3, 4. So that's the count of the number of errors. And so that was most of what I wanted to cover. Oh, I guess the one thing I did not show you, and so let me just point this out quickly for those of you who might be interested in this. That one thing that I thought was very interesting also with DataGraph is that if I export data that's in DataGraph correctly, then the exported data will not have those errors. And that is exported into Excel. So Excel can save this information properly. It's just that the individual cells have to be encoded as text. And in my DataGraph file, if I have my gene data, here it is here. Oh, actually, no, this isn't the, this is the gene data. Yeah, I think so. Yeah, this is it. So again, convert it back to text. All the original data is there, even though I did all those conversions. It's just interpreting the data. If I export this, then I can export it to an Excel file. You go under, let's see, where is it? File, export data. Notice it says selected columns only. So what you see here actually will change depending on what's selected. If I have nothing selected in the data table and I say file, export, now it says all visible columns because I haven't selected any in particular. It's just trying to give you some visual information about what it's actually going to export. But if I only wanted to export this column, I can say file, export data, and it would just export that, and I can, again, export that to an Excel file. And if I bring that data back into my spreadsheet, and I have an example where I already did it here, here's some test strings that are coming from data that I exported from DataGraph. And notice I'm double clicking on them right now, and that date field is not popping up because they're properly encoded as text. So this is an Excel file that you could share with others online and not be worried as being flagged as having errors in your data. Okay, well, I appreciate the positive feedback. I'm trying to think of what, we have some other topics that we have in the works. One is something that actually someone sort of requested to do a webinar on this before, about easy ways to, or how you bring data into the program, meaning replacing data. That's something that we're thinking about for next week. And so that would involve, copying and pasting in data. Obviously is just one very simple way to bring new data in, but also showing you how you can use DataGraph in scripting languages to create, graphs by using the dgraph command line utility. So, all the way from simple copy paste to more complicated, doing more complicated things. So that's one of the things that we're thinking about. And other than that, I appreciate everyone attending, and I hope you have a wonderful weekend, and we will see you next time.